Hello, my name is Shannon. This is Isaac, my genius son, and this is Train of Thought Science.
Train of thought science.
And tonight we're going to be talking about viruses and viral evolution, and viruses related to human evolution, and also Mollivirus Sibiricum. And also we're going to be talking about the function of viruses and how they invade a cell. Okay, Isaac, first.
Okay, so first I've got a picture of one. Crap. That is a real picture by an electron microscope of a T4 bacteriophage. The main difference between a microscope, compound light microscope and an electron microscope is that electrons are much smaller than photons, so they bounce those off of the object and then measure that. That's how they get so much smaller.
And describe the name of that particular virus and what that one attacks and so
It's
on.
called a T4 bacteriophage. Phage means to eat, and so bacteriophage, it does not invade people, just bacteria.
Bacteriophage.
Bacteriophage.
Bacteriophage. Next, we're going to talk about different types of viruses and their
So
structure.
do an introduction.
Okay, go ahead.
So we all know about viruses. They cause flu, common cold, but there is a lot more to it than that. For example, when you go swimming in the ocean, you're swallowing thousands and thousands of viruses. But the reason that you're not infected is because most of these are bacteriophages or something other than peoplephage, because that is totally a word.
Yeah, but viruses are very specific in what they attack or use to replicate themselves. Very specific.
And also, they only attack bacteria. That brings us on to a vocabulary term. Host range. HIV only infects immune cells, so it has a very narrow host range, as with something like the flu has a more broad host range because it infects more types of cells. But viruses are not considered living because for one, they don't reproduce, and for another, they're not technically categorized as a cell, which are two characteristics of all living things, and to be classified as a living thing, you have to fall under every single characteristic. So there are three types. There is three types of viruses. There is a helical virus, a polyhedral virus, and a complex virus. The helical virus, there isn't much to say about it. I'm going to zoom in on a picture.
Yeah, it's a spiral and it's stacked sort of like a DNA strand.
That right there is RNA. That goes through the capsid. Capsid is just a protective coating on a virus. And polyhedral viruses are mainly just a geometric shape. They can get up to 20 sides. That's the most complex that they can have. But they have extruding structures called glycoproteins, which act as keys, which we'll talk about later. You got it. And the complex virus, and this is really hard to understand. I cannot stress this enough. Complex viruses are the most complex. So, why do you keep doing that with your fingers? Okay, so it's the most complex, and on the bottom of it, they have the extruding tails of protein that help to lock on to the keyholes that we were talking about with the polyhedral virus. The T4 bacteriophage was a picture of a complex virus. That's a standard shape. Those are the protein tails.
Wait a minute. Well, let's zoom in on that. Show them the protein strands and where they key in at.
So they key in there. There are things on the surface known as antibodies, or antibodies actually attack the keys if they detect as a protein, or not a protein but a virus, so that the virus can't trick the cell. But onto the keys, over millions of years, viruses have evolved a way to get into the cell without them knowing. It's evolved a key, obviously, that the cell uses to identify proteins, such as simple monosaccharides, such as glucose or something. But some viruses have developed keys identical to certain types of protein, which tricks a cell into thinking they're protein. The T4 bacteriophage actually connects to these keys and just drills into the cell and releases its DNA, which we'll talk about what it does after that later. And then the polyhedral virus, the extruding structures on it are the keys, and it actually gets invaginated into the virus, and at that point it either releases its own genetic material or gets itself dissolved in the mitochondria or lysosome or wherever. Those are just organs in the cell. So once the DNA gets into the cell, it can enter two cycles, light tip or lysogenic. That's just an image. You know what? You can't read that. Okay. So, lysogenic is a more dormant cycle. It's where the DNA gets into the nucleus or the control center of your cell. Do you want to do some of this?
No, you're doing great. You're doing great. I'll interject with if I feel needed.
Okay, why don't you shoot and go get me water? Take my wallet, get me water. Okay, fine. Okay, so lysogenic cycle. It enters a nucleus and embeds itself into the DNA, the genetic code that all cells have. Never mind, I'm not gonna go there. And then once it gets into the code, the cell is replicated, and when a cell replicates, it replicates the DNA along with the tainted viral genetic information. A complex virus has DNA, a polyhedral virus has RNA, and a tobacco mosaic virus also has RNA. Not tobacco mosaic. I keep referring to the helical virus as a tobacco mosaic, but it's helical that I meant. Already knew all that? Okay.
He's saying it.
Okay, and once it replicates, it's in multiple cells, and there are a couple of stimuli that can activate the genetic code and command it to start replicating more viruses, but there's many stimuli too many to go into. But once it does that, the RNA or DNA gets out of the nucleus and goes to the ribosomes, which are responsible in protein synthesis, which is the production of proteins. And they get to the ribosomes, and the ribosomes make proteins, which are then transported to the nucleus and assembled into another virus. And from one virus's genetic information, it can create millions and millions more viruses, so many that the cell can't hold them all, and it actually bursts.
What's the other name for it?
Lyse, L-Y-S-E.
And I think they're two separate things. I'm not totally clear on the definition of them, but sometimes the cell will burst and then the cell will lyse, I guess, rupture without an explosion or something. I'm not sure.
So the lysogenic cycle is just a dormancy, and the lytic cycle is when it actually replicates. So a virus can either go straight to the light tip or lie dormant in the lysogenic cycle. Okay, on to our next topic, the Mollivirus Sibiricum, I think we should do first. You want to go with this?
I'm not going to be well spoken on it, but in the early 1900s, there was a sample taken from permafrost by scientists, and it actually sat in a isolated freezer for many years. It wasn't recognized as what it was. Scientists believed it was bacteria, but when it was later examined by, I can't remember the guy's name, but he recognized that it had viral structure to it, although it was very similar to a bacteria.
The reason that they mistook it as a bacteria is because it's actually giant. Standard virus is much smaller than any bacteria or cell. Okay, sorry about that. So back on the topic, I was describing the relation between the size of a standard virus and the size of this virus, the Mollivirus Sibiricum. A standard virus is maybe 1/1000th of the size of a bacterium or one of our skin cells, or any of our cells for that matter. But the Mollivirus Sibiricum is the same size, if not bigger than another. I'm going to go ahead and unlock this so I can show you the picture of the Mollivirus. It looks like a bacterium, and it even has a flagella, which is that tail right there. A flagella whips around and the medium that it's in to help it propel through the water or whatever it's in.
Similar to a sperm cell.
Yeah, I think that actually is what's on them.
Yeah.
Yeah. So it was discovered in 30,000-year-old Siberian permafrost near a squirrel's nest in the surface. So should we go ahead and describe the Mimi virus?
Yeah, go ahead. Mimi virus.
We didn't mention this at the beginning, but we'll also be describing the Mimi virus. Mimi stands for mimicking microbe. This is also a giant virus, but it mimics a bacterium so that another bacterium, such as an amoeba or paramecium, will consume it, during which it can get itself eaten and release its genetic code for replication. So Mimi, mimicking microbe. So now moving on to viruses and evolution. I think you're good with this one.
Yeah, there's been recent findings in science that there's a lot of evidence leaning or suggesting that viruses have strongly affected human evolution. Let's say a host gets infected with a virus, and the virus makes changes in their DNA during their lifetime. Yes, that happens, but the way that it can affect DNA is, let's say that the host is infected and transfers a sperm cell to an egg, and the DNA changes that are made within the sperm get into the egg and permanently get into the DNA code of subsequent generations.
I'm glad there's
And
no one eating here.
that happens enough times. Yeah.
Yeah,
If
we
that
were
happens
recording
enough,
in a
if that happens enough times, that permanently affects DNA, and which there's in DNA there's a lot of trash DNA, and scientists are now believing that most of this trash DNA comes from generations and generations and thousands and millions of times of DNA being changed by viruses. So a lot of this trash DNA is changes that were made by viruses. And, well, you can see where that can strongly affect human evolution.
This is an image of the Mimi virus. It looks like a polyhedral virus, so I'm going to assume that's what it is. Okay, so how about viruses being the first thing to exist?
I don't know how to speak on that, but there's...
Okay, I'll do it.
Okay.
So considering how some viruses such as the Mimi virus, not the Mimi virus, but the Mollivirus Ivericum and other similar viruses can now self-replicate, which as I said in the beginning of the video, viruses generally do not fall under that category. But this one can actually replicate itself. So considering the simplicity of it, it could have been one of the first organisms to ever exist billions of years ago. I wouldn't say billions, but maybe a billion years ago.
Especially since DNA is such a simple structure, such as RNA. If you have one of these self-replicating viruses that are made of a simpler form of DNA, RNA, it gives evidence and thought towards it possibly being an earlier organism than other organisms such as.
Mollivirus Ivericum.
Yeah.
Is that what you're going?
No, I was talking about science generally believes that other life forms came before viruses.
Alright. Okay, so DNA and RNA are actually pretty similar. They're both made of nucleic acid, which is just a compound in life. DNA is deoxyribonucleic acid, while RNA is ribonucleic acid. So they just drop the deoxy. So they're being pretty noisy. You think we should go and stop the video?
Did we speak on anything?
I think we did.
Okay. Well, we had a good video this time. We went through another previous on the same subject. Didn't go so well in a different setting. We decided to get back into restaurants. Better lighting. This one isn't particularly better lighting, but until our next talk, good night. Train of Thought Science.